Good morning, everyone. I hope that you woke up feeling great about yourself or feeling great about the things around you. But if not, there's still tomorrow, and I hope that you feel that way tomorrow, or the next day, and so on. So, for today's lesson, we will be talking about something that is somewhat very familiar to all of us, like literally all of us. So, if I ask you a question, why do you think that instead of listening to me physically or face to face, why are you listening at me and looking at me through your screen? Right? So you might have an idea as to what we're going to talk about right now, but I want you to save that idea because first we will talk about the objective of today's lesson. So the first objective is learn about the genetics of SARS-CoV-2 and how it differs from other coronaviruses. Second, learn about the mutation of the virus and the variants it produced. And lastly, learn about the molecular methods used to detect the virus and how it is performed. The unpredictable emergence of new infectious disease can be seen as threat to human health and global stability, despite extraordinary progress in development of countermeasures such as diagnostics, vaccines, and treatments. Diseases caused by coronaviruses are a few of many examples of emerging infectious diseases in the modern world. So, what is coronavirus? Coronaviruses are large envelope viruses with positive sense single-stranded RNA genome. The name coronavirus refers to the crown-like projections on the pathogen's surface. So, corona in Latin means halo or crown. And then researchers first identified the coronavirus in 1937 when they found out that some birds have bronchitis. While on the human coronaviruses, scientists found the evidence of human coronaviruses in the 1960s in the noses of people with common cold. So after hearing those years that are mentioned, we can say that coronaviruses are emerging and re-emerging pathogens. The emergence and re-emergence of some coronaviruses are believed to be because of the population shift from rural areas to urban areas and the increasingly frequent mixing of different animal species in densely populated areas. Today, coronaviruses are known as one of the most rapidly evolving viruses due to their high genomic nucleotide substitution and recombination rate. SARS viruses, for example, have the capacity to be directly transmitted from animals to humans. The of coronaviruses is also a result of their interaction with their host. For example, it was reported that the host shift of SARS-related coronaviruses or SARS-CoV mostly occurs in different species under the same genus, which is the Rhinolopus, indicating the genetic distance between hosts and also determines both the host shift and the cross-species transmission of the viruses. So we might already have an idea that there are different kinds of coronaviruses like SARS-CoV, MERS-CoV, and the like. But today, the human coronavirus that we will be talking about is the SARS-CoV-2, or the virus that is responsible for COVID-19, the global pandemic that we're experiencing right now. So, what is SARS-CoV-2? SARS-CoV-2 is a member of a large family of viruses called coronaviruses. These viruses can infect people and some animals. SARS-CoV-2 was first known to infect in people in Wuhan, China, and the virus is thought to spread from person to person through droplets released when an infected person coughs, sneezes, or talks. It may also be spread by touching a surface with a virus in it and then touching one's mouth, nose, or eyes, but this is less common. So as of October, around 243,822,145 cases are recorded, with 4,900,055 and 199 deaths. We all might be wondering, what does or how does coronavirus, the virus itself, looks like? So I'm going to show it to you. So what you're looking right now is the molecular structure of SARS-CoV-2, the virus that is responsible for the COVID-19 pandemic. The genome of the coronavirus codes four main structural proteins: the spike protein, or the S protein, the nucleocapsid N protein, the membrane M protein, and the envelope E protein, each of which play primary roles in the structure of the virus particle as well as in other aspects of the viral replication cycle. So let's first talk about the S protein. So this thing right here, kini siyang orange. That is the S protein or the spike protein. So the spike protein is responsible for allowing the virus to attach to the membrane of the host cell. It contains a receptor binding domain which recognizes a specific receptor, which is the angiotensin converting enzyme receptor 2, which is expressed in their lungs, heart, kidneys, and intestines. It has been shown that this protein binds to the ACE2 receptor with at least the same affinity and potentially as much as 20 times greater affinity than the SARS virus, which is the SARS-CoV that was detected around 2002. This could be one of the explanations for the reason why it's spreading so easily. The spike protein itself has two functional subunits, the S1 and the S2. The S1 binds to the host cell receptor and S2 mediates the fusion of the viral and cellular membranes. Because of the critical roles that the proteins play in binding to target cells and cellular entry, it is particularly focused on the design of vaccinations and medical treatments for COVID-19. So the next one is the M protein. So this thing is the M protein. M protein, or the membrane protein, is the most abundant on the viral surface and defines the shape of the viral envelope. It can be thought of as the central organizer for coronavirus assembly and interacts with other structural proteins. Now let's move on to the next protein, which is the E protein. So E protein, or the envelope protein, is the smallest of the major structural proteins on the viable membrane, which appears to have several roles. We know that it is integral in the assembly and release of the virus from host cells and during viral replication. It is largely localized at the site of intracellular trafficking, more specifically at the endoplasmic reticulum and the Golgi apparatus. So essentially, the M and E proteins play a critical role in turning the host cell apparatus into workshops where the virus and our own cells work together to make new viral particles. After this, let's talk about the viral envelope. Underneath all this protein, there's this thing called the viral envelope. This is isolate the So this thing is the viral envelope. This is the virus's outer layer and is derived from the host cell membrane, like ourselves or the animals. It's a fatty layer and worth noting that in contact with soap, it will break down, killing the virus. And this is the reason why hand washing with soap is so important to prevent the spread of this virus. So guys, always remember to wash your hands, okay? So now we will go sa insides sa atong virus. So na yun na siya sulod, guys. Sulod ana is, you can see the N protein and the RNA. So this is the N protein, or the nucleocapsid protein. The N protein appears to be multifunctional in particular. It essentially inhibits a lot of host cell defense mechanisms and assists the viral RNA in replicating itself and therefore creating new particles. So let's move on to the RNA, which is inside the nucleocapsid protein. So the RNA, or the ribonucleic acid, it is where all its genetic information is held to allow itself to replicate. So the coronavirus genome plays multiple functions during viral infection. It acts initially as an mRNA, which is translated into two large replicase polyproteins. And in fact, these polyproteins are the only translational products derived directly from the genome. And that's all for the structure of the SARS-CoV-2, and I hope that you have learned something from this discussion. Thank you so much for listening.
Okay, so let's move on to the emergence of coronavirus diseases. So when we say coronaviruses, they are emerging and re-emerging pathogens. So when we say pathogens, no, they are the organism that causes diseases. And several of them have caused serious problems in both humans and animals. So this includes varying symptoms ranging from mild respiratory illness to severe infectious causing death. So apart from the respiratory tract, coronaviruses can also affect other organs of humans or animals in the body, such as intestinal tract, liver, kidney, and even your brain. So there are different coronaviruses. So the alpha, the beta, the gamma, and the delta coronaviruses. Okay, so we're going to talk first about the alpha coronaviruses. So there are actually two alpha coronaviruses now that causes illness in human. So as you can see here, the first one is the HCOV229E or the 229E coronavirus. So it was discovered in the mid-1960s and it's more likely infect mga immunocompromised patients. But as you notice that this term is not that familiar with us compared to the SARS, MERS, and the COVID-19 because it is less symptomatic no, or less severe than the other coronaviruses. And the second alpha coronaviruses that affects humans is the HCOV NL63, which was discovered in 2003 in Netherlands. So it is the second most common virus since it infects no humans like children. But it depends in season, like the case is high in winter and the case is low in summer. So these viruses, both of them, causes common colds, acute rhinitis, stuffy nose or runny nose. It can also cause pneumonia, vomiting or diarrhea. And the group of people who are at risk are the young children, the older adults, and the people with disability or those PWDs. So the next group that we're going to talk about are the beta coronaviruses. So it actually have three lineage, the lineage A, the B, and the C. So let's go on first with the lineage A. So there are two beta coronaviruses under it. The first one is the HCOV OC43 or the OC43 coronavirus, which was discovered in 1967 in a laboratory in Maryland, United States. So actually, this is said to be the most common strain of coronaviruses that causes infection. But among all lang, the HCOV, except for SARS, MERS, and the SARS-CoV-2. Then the second coronavirus is the HCOV HKU1 or the HKU1 coronavirus, which was discovered in 2005 in a laboratory in Hong Kong. That's where it's associated with a febrile seizure, particularly in children. So both of these viruses also have a worldwide distribution, like they are everywhere, like those other two alpha coronaviruses that we talked earlier. It also causes colds, lower respiration, and gastrointestinal symptoms. Then in lineage B, so there are actually two beta coronaviruses now, the SARS and the SARS-CoV-2. So the SARS first, so the first one is the SARS-CoV or the SARS coronavirus. This coronavirus now was officially discovered in 2003, but likely started in 2002, and it was originated in the Guangdong province of China. But with the concerned efforts of the global community, fortunately, this coronavirus was expelled from the human population in July of 2003. So basically, it had an outbreak from February of 2003 to July of 2003. So it means it has no short period of time, the outbreak, compared to what we're experiencing now. Now it's almost how many years? So what is believed to have happened here is that the virus seems not to have spread from a bat to a civet. So if you don't know, this animal is a small lane and is a mammal native to tropical Asia and Africa. This is actually looks like a cat, but it's not a cat. Then a civet to the human. So this virus causes condition known as severe acute respiratory syndromes or SARS, which causes severe symptoms like fever, cough, chills, achy muscles, and it can ultimately lead to acute respiratory distress syndrome, which can lead to fatality. And in fact, the basic reproduction number of SARS is anywhere from 0.7 to 3. So it means one infected individual can infect 3 other people. Aning a SARS. Then the next one is the SARS-CoV-2 or known as the coronavirus 19. So as what I said earlier, it is discovered in late 2019 in Wuhan, China, and was been a worldwide pandemic starting from February to March of 2020. So it is said that it came from a bat and was transmitted to human. And just like the first SARS, it can cause acute respiratory distress syndrome and fatality. So as you can see in the information below, there are more than 1.3 million cases with 75 deaths. But that was a year ago information. As of right now, it's over 243 million and a total of 4.94 million deaths. So after the lineage B, we go on to lineage C. So in lineage C of beta coronaviruses, there is this MERS coronavirus, or what we call the Middle East Respiratory Syndrome coronavirus. So this was discovered in 2012 in Saudi Arabia, And it really hasn't gained that much attention out of the world because it's only been localized in Saudi Arabia. And on the outbreak that was happened in South Korea. So this is said that it came from the camels or the milk camels, and where if an individual drink the camel milk, then that individual will be exposed to that virus. And this condition is very similar to SARS now because the infections can be anywhere from asymptomatic to severe and fatal, like fever, cough, shortness of breath, severe pneumonia, and acute kidney injury. Fortunately lang, the basic reproduction number of MERS-CoV is actually very low. It quoted to be in 0.3 to 0.8. So the basic reproduction number is less than 1. So it doesn't spread to a lot of people because the virus eventually dies out. So as of this moment, and since the virus is still in the world, it has more than 2,400 cases and a total of 858 confirmed deaths. Facts. So they actually have similar RBD structures to the SARS-CoV, the SARS-CoV-2, and the SARS-CoV and the MERS-CoV-2. But unlike SARS, MERS used lang the DPP4B propeller as a receptor. So as we can see, the key features of the SARS-CoV, the MERS-CoV, and the SARS-CoV-2 or the COVID-19 are all the same, that they exhibit dominance of hospital-acquired infection and pathogenesis driven by a combination of viral replication in the lower respiratory tract and then aberrant host immune response. So these emerging viruses, these beta viruses, are zoonotic, or there is a cross-species transmission, which means these viruses can be transmitted from the animals to humans. Thus, we can say that the SARS-CoV, the MERS-CoV, and the SARS-CoV-2 or the COVID-19 are the three highly transmissible pathogens that emerged in humans over the past 2 decades. And the next one would be the one of the general coronaviruses, which is the gamma coronaviruses. So this virus was actually discovered in Brazil, and it was first spotted in the Amazon city at around December of 2020. So it caused a lot of problems during that times because it has contributed to a surge in cases. Thus strained the Brazil's health system and even led to oxygen shortages. Thus, it affects a lot of people, and it also has been reported in at least 72 countries. And lastly is the Delta coronaviruses. So this virus is actually known as the fast-spreading virus that stoked a dramatic wave of COVID cases. Now it surged the case in India because it was first discovered there, and it overwhelmed a lot of hospitals, and it has been found for at least 96 countries, including our country, the Philippines. This virus is also known as to be 55% more transmissible than the Alpha coronavirus. Okay, so to have an in-depth knowledge about these viruses, mutations, and variants, here are Joanne and Ivan to discuss it.
So I'm going to tackle about the mutations of the virus and the variants it produced. So, since the commencement of the COVID-19 pandemic, genetic lineages of SARS-CoV-2 have emerged and circulated all over the world. So, in the United States, SARS-CoV-2 genetic lineages are continuously tracked by epidemiological investigations, viral genetic sequence-based monitoring, and laboratory research. So, there is this group in the US government, which is called SARS-CoV-2 Interagency Group, that created a new class of SARS-CoV-2 variations. So there are 4 categories: variants being monitored, variants of interest, variants of concern, and variant of high consequence. Before I start defining each type of variants, I'm going to define first what is mutation, lineage, and variant. So when we say mutation, it refers to a single change in a virus genome. So mutations happen very frequently, but only sometimes change the characteristics of the virus. And when we say lineage, it is a group of closely related viruses with a common ancestor. So as we all can see, SARS-CoV-2 has many lineages. And variant is a viral genome that may contain one or more mutations. So, how are variants classified? So, the SIG meets on a regular basis to assess the danger presented by SARS-CoV-2 variants circulating in the United States and to provide categorization recommendations. So, this assessment is carried out by a group of subject matter experts who look at data such as variant proportions at the national and regional levels. So, what do you mean by variant being monitored? So, these are variants for which there is data indicating a potential or clear impact on approved or authorized medical countermeasures. And when we say variant of interest, it is the specific genetic markers that has been linked to changes in receptor binding, decreased neutralization by antibodies generated in response to previous infection or vaccination. And when we say variants of concern, with evidence of increased transmissibility, more severe diseases, significant reduction in neutralization by antibodies generated during previous infection or vaccination. And lastly, for variant of high consequence, a high impact variation has clear evidence that preventive strategies or medical countermeasures are much less effective than previously circulating variants. So you can see a table there. It is categorized by the SIG with its variants and its categorization. So let's now move for each variant and their mutations. So alpha, the alpha variant. So we already know that this virus was discovered in the United Kingdom and it has already spread to over 50 nations. Since November 2021, the dissemination in this variation has resulted in an increase in COVID-19 cases in the United Kingdom, and at least 140 nations have already detected the variation. So when we say its mutation, in both the hamster model and primary human airway epithelial cells, the 501Y mutant virus demonstrated consistent fitness advantages for replication in the upper airway. So the 959, the N501Y mutation in spike protein's receptor binding domain can increase its affinity for the human ACE2 receptor. So, a major impact on fast spread of the variant. So the P68-1H mutation has the potential to impact viral infectivity and replication in cells. So as you can see in the picture, and the UK variant has given rise to a new variety, the Bristol variant, which carries the E484K mutation, possibly encouraging immune response evasion, which is also seen in the African and Brazilian variants. So, a B117 sublineage evolved as a result of the consecutive acquisition of a V70L mutation in the membrane protein, followed by a unique D178H mutation in the S protein. So the D178H mutation in the S protein was discovered to be in the N-terminal domain of the S protein, near to two additional characteristics mutation of B117, HV6970del, and Y144del. It is surface exposed and has the ability to modify the N-terminal domain tertiary conformation or accessibility, hence affecting neutralization by anti-D-directed antibodies. So you can see in the picture the 3D structure of the spike protein of the UK variant, mutations in different color. So for the beta variant or the B1351, the virus was first discovered in South Africa. It has already been diagnosed in at least 20 additional nations, including the United Kingdom. So this variation arose with extremely high number of mutations. So its mutation, notable mutations include N501Y, K417N, and E484K in the spike protein's receptor binding domain, which can increase its affinity for the human ACE2 receptor, which has been explained in alpha variant. So the E484K mutation may result in immune response escape. So you can see the 3D structure of the spike protein of the South African variant, which is called the beta variant. So for the Delta variant or the B.1.6172, it is first identified in India. So the variant could be more than twice as transmissible as the original strain of SARS-CoV-2. So its mutation 19R 156 del, 157 del, R158G, L452R, T478K, D61, and other mutations in the spike protein identified the variant. So several of these mutations may have an influence on immune response aimed towards the receptor binding proteins important antigenic areas. So as well as the loss of a portion of the N-terminal domains. So the P681R mutation alters an amino acid right next to the furin cleavage site, a crucial step in allowing the virus to penetrate human cells and thereby increasing viral infectivity. For the Gamma variant or the P.1, it was first discovered in Brazil and now spread to over the nations including the United Kingdom. In the city of Manaus, it has caused widespread infection. So its mutation, the receptor binding region of the spike protein, contains the amino acids N501Y, K417N, and E484K, which can improve its affinity for the human ACE2 receptor. The E484K mutation may allow an individual to evade the immune system's reaction. So as you can see, there is a 3D structure of the spike protein of the Brazilian variant there, with its corresponding different mutations.
The Epsilon variant was first detected in California, USA, and has two distinct lineages, the B1427 and B1429. The lineage B1429 is characterized by 5 mutations, the I4205V in RF1A, D1183Y in RF1B, S13I, W152C, and L452R in the S protein. The L452R mutation in the S protein defines the B1427 variation. The L452R mutation may improve interaction between the spike protein and its human ACE2 receptor, increasing its infectivity. The ATA variation in lineage B1525 is also known as BUI-21Feb-03 by Public Health England. It's a SARS-CoV-2 variant. It does not have the same N501Y mutation as alpha, beta, or gamma, but it does have the same E484K mutation as gamma, zeta, and beta. In December of 2020, this variety was first discovered for the first time in Nigeria and the United Kingdom. It is also seen in other countries including US, Canada, Denmark, Britain, France, Belgium, Spain, Finland, Nigeria, Ghana, Jordan, Australia, and Singapore. And 3 mutations define this variant: E484K, Q677H, and F88L. Other changes make it resemble the UK version, the B177. The biological properties of this variation, such as its infectivity, are still unknown, and is presently being researched. The Utah variation of SARS-CoV-2, or also known as lineage B1526, is one of the virus types that causes COVID-19. It was initially discovered in November 2020 in New York City. By February 2021, it had quickly expanded throughout the New York area, accounting for almost 1 out of every 4 viral sequences. The E484K spike mutation, which may help the virus evade antibodies, and the D2K3G occurs in the antigenic supersite within the N-terminal domain, have been found in at least 48 US states and 18 nations by April 11, 2021. The Kappa variant, which has mutations in the spike proteins, is considered a highly transmissible SARS-CoV-2 variant, and it was initially found in India. The spike protein of Kappa contains 4 mutations, the E154K, L452R, E484Q, and P681R. It's unclear whether this variant's entry efficiency, host-tropism, or sensitivity to neutralizing antibodies and entry inhibitors has altered. Furthermore, the P618R mutation causes increased spike cleavage, which may make viral entrance easier. B6173 is a lineage of SARS-CoV-2 B617. It was discovered in January 2021 and contains a variety of mutations in the SARS-CoV-2 spike protein's N-terminal domain and receptor binding domain that may boost the variant's immune evasion potential. Sub-lineage B1673 has 9 to 10 spike mutations and 2 receptor binding domain mutations, the L452R, and 7478K. It is most common in the countries of Turkey, India, United States of America, Angola, and United Kingdom. One of the forms of SARS-CoV-2, the virus causes COVID-19, is the Mu variant. It is also known as lineage B1621 or VUI-21 It was initially discovered in Colombia in January 2021. And on August 30, 2021, the WHO identified it as a variant of interest. And according to WHO, the variation features mutations that signal a risk of resistance to current vaccines, and that more research is needed to fully understand it. The Mu variety has caused outbreaks in South America and Europe. Zeta variant, also known as lineage B.2, is a variant of SARS-CoV-2, the virus that causes COVID-19. It was first detected in the state of Rio de Janeiro. It harbors the E484K mutation, but not the N501Y and K4117 mutations. It evolved independently in Rio de Janeiro without being directly related to the Gamma variant from Manaus. The Zeta genome has 3 amino acid mutations: E484K, D614G, V1176F, all of which are found in the virus spike protein code. And according to the Centers for Disease Control and Prevention, E565L has been detected in some of the Zeta variant sequences, but not all. Various mutations are nothing new. SARS-CoV-2, like all other viruses, evolves over time. The majority of these modifications are minor, but others have the potential to affect the nature of these viruses, making them more aggressive or allowing them to evade treatment or vaccines.
COVID-19 is spreading every day like a fire across the globe with infection symptoms and many common ailments. Therefore, specific diagnosis is crucial for further management and to prevent spreading of infection. So actually, there are different types of tests that are available for the detection of COVID-19 infections. First is a direct test to diagnose the viral infection, which is the RT-PCR test, while for the indirect tests such as antibody test for checking immunity and surveillance, and antigen test to detect specific viral proteins.
Now let's talk about the RT-PCR. So to perform this test, the samples can be collected by a nasopharyngeal swab or an oropharyngeal swab for sa ato barong ilong or sa atong baba. So for the nasopharyngeal specimen, the swab is inserted in the nostril and gently moved into the nasopharynx, then rotated for a specific period of time to collect secretions that contains the virus. So after that, the swab is placed immediately into the sterile tube containing a vital transport medium. So dito sa sterile tube, ilan na silang i-secure for ready for testing na. So polymerase chain reaction (PCR) is a standard method for coronavirus testing, and it is widely used in molecular biology to make millions or even billions of copies of a specific DNA fragments. So for the coronavirus or the COVID-19, wala na siya contain of DNA, but rather it contains a long single-stranded RNA genome. So to detect these viruses with PCR, RNA molecules must be converted into their complementary DNA sequence by reverse transcriptase. Then, the newly synthesized DNA can be amplified by standard PCR procedure.
To perform this RT-PCR method, basically, viral RNA should be extracted. So to extract the viral RNA, a commercial kit is used. Then, the extracted sample is first added into the microcentrifuge tube. Then, mixan siya og lysis buffer. This buffer is, pertaining to the lysis buffer, this buffer is highly denaturing. So once the lysis buffer is added, the tube is mixed by post-vortexing and incubated at room temperature. So once the sample is lysed, a purification procedure is carried out by using a spin column. So the sample is loaded into the spin column, then a centrifugation is performed. So this procedure is a solid phase extraction, in which the stationary phase consists of a silica matrix. So under optimal salt and pH condition, RNA molecules are bind to the silica gel membrane, and at the same time, protein and other contaminants are not retained.
After na po sa centrifugation, the spin column is placed into the clean collection tube and the filtrate is discarded. So after i-discard ang filtrate, i-addan siya og wash buffer. So the column is put in the centrifuge again, forcing the wash buffer through the membrane. So this buffer removes any remaining impurities from the membrane, leaving only the RNA bound to the silica gel. So once the sample is washed, the column is placed in a clean microcentrifuge tube, and then another buffer is added, which is the elution buffer. Then a centrifugation is carried out, forcing the elution buffer through the membrane. The elution buffer removes the viral RNA from the spin column, and a purified RNA, which is free of protein, inhibitors, and other contaminants, is obtained. After the extraction of the viral RNA, the next step is the preparation of the reaction mixture for the PCR amplification. In this step, a master mix is used, which is a premixed concentrated solution that consists of buffer, reverse transcriptive enzyme, nucleotides, forward primer, reverse primer, Tachman probe, and DNA polymerase. Finally, to complete this reaction mixture, the RNA template is added. The tube is mixed by Pohl's vortexing. Then the reaction mixture is loaded into the PCR plate, which generally contain 96 wheels, allowing the analysis of several samples at the same time. Next, the plate is placed in a PCR machine, which is essentially a thermal cycler. Real-time RT-PCR is used for the detection of the new coronavirus 2019 by the amplification of target sequence in the RD/RP gene, the E gene, and the N gene. The choice of target gene depends on the primers and the probe sequences. The first step in RT-PCR is reverse transcription. The first-strand complementary DNA synthesis is primed with a PCR reverse primer, which hybridizes to a complementary part of the virus RNA genome. Reverse transcriptase then adds DNA nucleotides onto the 3' end of the primer, synthesizing DNA complementary of the viral RNA. The temperature and the duration of this step depend on the primer, the target RNA, and the reverse transcriptase used. Next, an initial denaturation step is applied, causing denaturation of the RNA-DNA hybrids. This step is required for the activation of DNA polymerase and simultaneously the inactivation of reverse transcriptase. PCR consists of a series of thermal cycles, with each cycles consisting of denaturation, annealing, and extension steps. The denaturation step consists of heating the reaction chamber to 95 degrees Celsius, and it is used for denaturation of the double-stranded DNA template. In the next step, the reaction temperature is lowered to 58 degrees Celsius, allowing annealing of the forward primer to its complementary part of the single-stranded DNA template. The annealing temperature relies directly on the length and composition of the primers. In the extended step, the DNA polymerase synthesizes a new DNA strand complementary to the DNA template strand by adding free nucleotides from the reaction mixture that are complementary to the template in the 5' to the 3' direction. After the first cycle, the double-stranded DNA target is obtained. Then the denaturation of this double-stranded DNA is performed, yielding 2 single-stranded DNA molecules. In the next part, the reaction temperature is lowered, allowing annealing of the primers to each of the single-stranded DNA templates and annealing of the Tachman probe to its complementary part of the target DNA. Tachman probe consists of a fluorophore attached to the 5' end of the oligonucleotide probe. The fluorescence is emitted by the fluorophore when it is excited by the cycler's light source. Also, this probe consists of a quencher at the 3' prime. In the extension step, DNA polymerase synthesizes new strands. When the polymerase reaches a Tachman probe, its endogenous 5' nucleus activity clips the probe, separating the dye from the quencher. With each cycle of PCR, more dye molecules are released, resulting in an increase in fluorescence intensity proportional to the amount of amplicon synthesized. This method allows the estimation of the amount of a given sequence present in the sample. The number of double-stranded DNA pieces is doubled in each cycle. Therefore, PCR can be used to analyze extremely small amount of sample. For the measurement of the fluorescent signal, a tungsten halogen lamp, an excitation filters, mirrors, lens, and emission filters, and a charge couple device, CCD camera, are used. Filtered light from the lamp is reflected off mirror, passes through a condensing lens, and is focused into the center. Then passes through an emission filter and is detected by the CCD camera. In each PCR cycle, light detected by the CCD converts the light that it captured into digital data. This method is known as real-time PCR, which allows the monitoring of the progress of the PCR reaction.
Why choose real-time RT-PCR tests? RT-PCR is considered as the golden standard for diagnosing the coronavirus. It is one of the fastest and most accurate laboratory methods for the detection of the COVID-19 virus.
The RT-PCR test is highly specific and a positive test result is highly confirmatory as dual tests are done before reporting, first for the screening and secondly for the confirmation. When a person is exposed to a new virus and after an incubation period, the immune system creates proteins called antibodies as a part of the body's natural response to fight the infection and to neutralize the pathogen. Antibodies can come in different varieties known as isotypes or classes. IgM antibodies are produced against the viral antigen in the early stages of the infection and are detectable after 4 to 7 days after symptoms develop. Following that, IgG antibodies are produced 7 to 14 days after the onset of the symptoms and are detectable for months and even years, depending upon the antigen and the individual. These antibodies can also be detected if a person has an infection without any symptoms. The COVID-19 antibody test is used to detect the presence of the anti-SARS-CoV-2, IgM and IgG antibodies in the human whole blood, serum, or plasma samples by using a cassette-based system. To perform a test, the cassette is placed on a level surface at room temperature. Then one of the fingers is punctured with lancet and a plastic dropper is used to take several drops of blood. Then one drop is added into the cassette sample well. Finally, drops of a buffer are added into the sample well to facilitate the test, and the cassette is incubated for 10 minutes. The test kit is a qualitative lateral flow immunochrographic assay, and the test device contains a sample pad where the combined sample flows down a conjugation pad, which contains gold COVID-19 antigen conjugate. The antigen will then bind to the anti-SARS-CoV-2 antibodies in the sample, and two different antigens can be used with affinity to IgM and the second with affinity to IgG antibodies. The virus antigen is labeled with gold nanoparticles which can be used as a visual indicator because of the nanoparticles' interactions with visible light, so that the gold nanoparticles absorb and scattered light, resulting in a red color. In addition, rabbit IgG antibodies labeled with gold nanoparticles exist in the conjugation pad, and they are used for quality control to assess the test validity. The test strip also contains a nitrocellulose membrane with 3 lines. First line coated with anti-human IgM antibodies, second line coated with anti-human IgG antibodies, and the third line coated with anti-rabbit IgG antibodies. In addition, an absorption pad is used in the test strip to move the sample across the test. The sample migrates laterally across the membrane by capillary action. During the testing, the antibodies in the sample reacts with the SARS-CoV-2 antigen, and it conjugates gold nanoparticles in the conjugation pad. The mixture then migrates laterally across the membrane by capillary action, and the IgM and IgG antibodies are bound to the anti-human IgM and anti-human IgG antibodies respectively, while the rabbit IgG antibodies are bound to the anti-rabbit IgG antibodies. Only those conjugated with COVID-19 antigen gold nanoparticles complexes will produce a visible colored line. The test result is positive if both IgM and IgG antibodies are detected, if only IgM antibodies are present, and if only IgG antibodies are detected. When IgM is positive and IgG is negative, the subject's immune system is actively producing antibodies to arrest an infection with the target virus. When both IgM and IgGs are positive, the subject's immune system is actively producing antibodies to an ongoing infection that likely began more than 14 days ago. When IgG is positive and IgM is negative, the subject's immune system has produced antibodies to a target viral antigen. A negative test result means that the person is not infected with the virus, or perhaps the person has a recent infection but the antibodies are not yet produced. Also, since the accuracy of the test depends on the amount of the antibodies, some people may not develop detectable antibodies after the infection. Since the COVID-19 is a new disease, we don't know exactly how long the antibodies last, so a negative test results could mean that the person has been infected in the past and the antibodies are no longer detectable. The test result is invalid if the quality control line C does not show a red color. Therefore, the samples must be retested. Antigen rapid tests are an important pillar during the COVID-19 crisis. In contrast to a PCR test, antigen rapid test can show whether a patient is positive or negative in about 15 minutes. Most of them are based on the so-called lateral flow test similar to a pregnancy test. Rapid tests are intended to detect the presence of the substance of interest for the COVID-19. This might be a surface protein of the coronavirus. So the sample is pipetted onto a defined spot and through capillary flow, the sample runs along the surface of the pad. Here, a visible result is shown of the test and control lines. Two lines signal that the sample was positive, whereas the line alone means negative. When looking at the molecular mechanisms, antigen rapid tests are based on antibodies. Three different variants are used here, starting with the primary antibodies, which are conjugated to particles. They are, for instance, labeled with gold labeled particles. These antibodies can specifically bind a surface protein of the virus. The second set of antibodies does the same, but they are immobilized on the test pad. The third set of antibodies are secondary antibodies, which can bind to a part of the primary antibody when placing the sample containing virus material onto the test pad. The labeled antibodies bind the surface marker of the virus capillarily. Flow lets the fluid run along the test. Here, the primary antibodies in blue will also capture the virus. Parts of the sample which are not captured here will run and pass the line of secondary antibodies. They can capture the labeled primary antibodies. The label is visible as a red line later. The combination between the visible test line and the control line signals as the sample was positive. In case a sample does not contain any virus, the first line of primary antibodies in blue cannot capture viral proteins. That also means no labeled antibody will stick here. The secondary antibodies, however, will bind the mobile antibody. Hence, we have a signal for the control line but not the test line, which means the samples was negative.